Hi, you're back with me. Yes, you know me. I'm Professor Kukreja. So yeah, let's go. It's according to the HGP, yeah, the Human Genome Project, right? Started in 1990. Yes, a new branch we know it came up with in the similar era, that was bioinformatics, right? Applying a lot of computers and biology together, right? That gave rise to this bioinformatics. Anyways, 1990 में शुरू हुआ ये project, Human Genome Project. Total number of genes in humans genome was around 30,000. Yes, earlier they estimated that it could be around 80,000 to 1,40,000 genes. But now we know that we have approximately 30,000 genes, right? So the number of genes on the Y chromosome, yes, one of the chromosome with the least number of genes, right? In the entire human genome of 3164.7 million bases, right? This entire genome, we know that they have different gene sizes, right? A typical average gene has a size of around 3000 bases, right? And the largest one, yes, you know that is a dystrophin with around 2.4 million bases, right? So with these 30,000 genes, as I told, out of which like we all have around 99.9% similarity with all of us, we know that out of all this, the entire human genome of which I just mentioned, 3164.7 million base pairs, less than 2% is actually coding for what you call as proteins, and we actually don't know actually the functions of approximately about 50% of the entire discovered genes, right? They have a lot of these repetitive sequences, some repeated around 100 to 1000 times, but then they don't code for proteins maybe up to now, our knowledge up till now, but they are important in structure, in dynamics, in evolution, right? With around, I suppose, 1.4 million locations which have this SNPs, the single nucleotide polymorphism. SNPs, what you call, SNPs, what you call. So here, the number of genes on Y chromosome, the shortest one, you know, the what you call as one with the maximum genes is the one with 2968. Yes, chromosome number 1, right? And the smallest one is the chromosome Y, which has the genes of 231. Oh, I don't have a chalk here, but you remember it is the answer of 231. Anyways, in eukaryotes, right, the anticodon of tRNA that pairs with the start codon of mRNA. Let me get a chalk here, right? So, sorry. So, the first codon on mRNA, whether pro or eukaryote, it's AUG, right? And we want the anticodon on tRNA, right? So the tRNA, it has anticodon which is complementary to the first codon, coding for amino acid, you know, methionine, right? So in prokaryotes, it's N-formal methionine, and here it's methionine, right? So AUG, so complementary to AUG would be UAC. Can you find UAC here? Yes, we have UAC, and here we have this 231. So stay tuned with me, Professor Kukreja, for more on NEET biology. Thank you.